Okay, I'm going to try to finish up with synapses here, finishing up talking about the chemicals, and then getting into the types of synapses. And I'm providing this picture that I got from another textbook that is showing the areas of the brain to which dopamine flows, so the dopamine pathways, and it's also showing the serotonin pathways. And you can see that there is some overlap there in the pathways, but they're also going to very different regions, as the dopamine is going up through the striatum, that's controlling the movement, and then from the ventral tegmental area to the nucleus accumbens, which is that really influencing the reward center, and going up into the prefrontal cortex, which is really helping us with, if it's healthy, we are not kind of more impulsive and so forth. We're inhibiting some of our impulses. And then serotonin going up through some of those subcortical areas that we see from the raphe nuclei, but also up through the many different areas of the cortex. And one of the reasons I leave this slide in here is to talk about the relationship between dopamine and serotonin. So there's really kind of a balance that exists. One of the ways we also can cause ourselves some depression is by getting too much of the phenylalanine or tyrosine in our diets, as those are larger amino acids, and they I want to say they kind of bully their way across the blood-brain barrier at the expense of tryptophan. There was even one study where they gave young college students, young males, either a diet that was very high in tyrosine, so they gave them corn and aspartate, so Diet Coke, and those young men were more likely to get into arguments and to actually become violent with each other and aggressive with each other than young men who were given a different diet. So we can lower our serotonin in other ways, and there is this kind of balance. And what this makes is one of the problems we might see is if we have some depression and we have attention deficit disorder, attention deficit hyperactivity disorder, that raising that serotonin with a selective serotonin reuptake inhibitor can lower the dopamine to a point where we become really overly impulsive that it's problematic. So these are some of the things that we have to keep in mind as we're prescribing drugs, that there are these relationships between, and often a balance between, many of these different neurotransmitters. Dopamine also has its balance with acetylcholine, as we'll see eventually down the road, as well as with glutamate. I'm going to talk briefly about nitric oxide. If you remember, it is it's a gasotransmitter, and it is released oftentimes. It is the negative feedback that is sent from the postsynaptic neuron to the presynaptic neuron, letting the presynaptic neuron know, hey, you've already sent me a message, and inhibiting further communication. It's also a gas that's released by small local neurons when they're active, and so there's more nitric oxide in these areas of the brain where there's high activity, which also allows for the dilation of blood vessels and more blood to flow to that area, because we're going to need more blood, we're going to need more oxygen in an area where we just used the oxygen. Those neurons are using oxygen out of the blood. Most neurotransmitters are stored in synaptic vesicles. Nitric oxide is not. Again, it is a gas. Those vesicles, we have a lot of them in the presynaptic terminals. Neurons can accumulate excess levels of neurotransmitter in those presynaptic terminals, and so neurons have a way to stop that accumulation. So I have the example here: neurons that release serotonin, dopamine, or norepinephrine, they all contain this monoamine oxidase, sometimes shortened to MAO. That enzyme breaks down those neurotransmitters and makes them inactive, so that stops this really excess of accumulation of the neurotransmitters. So one of the older antidepressants that does not get used very often anymore, but if it's sometimes used as a last defense if we've tried many antidepressants with someone and nothing is working, is an MAO inhibitor. So it's inhibiting this enzyme, and it's allowing a little bit more accumulation of those neurotransmitters because what we're assuming is that's where the issue lies. They don't have quite enough serotonin, dopamine, or norepinephrine. And as I kept saying with that simple picture of one neuron communicating to another neuron, and I kept saying it's more complicated, it's more complicated. So some of the complication is not just in, right, that I'm communicating to many, many neurons, and many, many neurons are communicating to me, most likely. But so another piece of this is that we're probably most neurons are releasing a combination of more than one neurotransmitter, so 2 or more neurotransmitters. And at least one kind of neuron releases different neurotransmitters from different branches of its axon. So I have an example here of motor neurons. They have one branch that's going to the muscles, and as I mentioned, it's always between a motor neuron and a muscle. We are releasing acetylcholine. They have another branch coming off the end of the axon there to communicate to other spinal cord neurons, and those branches are releasing acetylcholine and glutamate. So that's another kind of level of just complexity. So I, as a receiving neuron as well, I can be responding to many different neurotransmitters at these different synapses, all at the same time as part of that neural integration. And so to a large extent, what we're doing with neurotransmitters, with these excitatory and inhibitory messages, they are like the ones and zeros in our computers, right? We can do a whole lot with simply on-off messaging, but messages are actually more complicated and varied than just on-off. So when a neurotransmitter attaches to a receptor site, the receptor may open a channel, and I'm going to talk about this for a little bit here, which are called ionotropic effects. They are allowing in ions. Or it may produce a slower but longer lasting effect, which are called metabotropic effects. I'm going to talk about that as really kind of changing the metabolism of the cell. So I'm first going to talk about the ionotropic effects, which is how we're just influencing ion flow. Are we allowing those ions to flow into a neuron, or are we closing off the gate and stopping those ions from flowing? So this is the opening and closing of these ligand-gated channels, as they're called, where ligand is a word meaning chemical. Sometimes they're called transmitter-gated channels. So those neurotransmitters are chemicals, and again, we can have the same effect as one of the neurotransmitters by taking a particular psychotropic drug. So it's doing the same kind of ligand-gated, chemical-gated work, or we can oppose that particular neurotransmitter, if you remember from my very short stint of talking about drug effects. These occur very quickly. They occur within 1 millisecond with a half-life of 5 milliseconds. So they happen and then they stop. This is really important for any kind of information processing where the speed of updating that information is important. So visual and auditory information, we need that information. We need to deal with that information very quickly. And so these are ionotropic effects on these neurotransmitters. I have written here most excitatory ionotropic synapses are glutamate. If you remember, I said that's a widely distributed excitatory neurotransmitter. And most inhibitory ionotropic synapses are GABA, my widely distributed inhibitory neurotransmitter, GABA, gamma aminobutyric acid. Those are just the most common of those, but we also see other neurotransmitters having this ionotropic kind of effect. So just as examples of 2 more neurotransmitters that have ionotropic effects, glycine in the spinal cord is an inhibitory neurotransmitter that has an ionotropic effect. It really stops us from contracting muscles too much. Acetylcholine is often, and in most cases, it's an excitatory ionotropic. So those nicotinic receptors are having an ionotropic effect. And that's what's really being shown in the picture there. And part of the reason I bring up acetylcholine is to talk about this picture where if we look at the ion channel and we see it in A, that kind of outer portion, the red portion is embedded in the membrane, and the inner portion is a sodium channel. But when you look at it A in comparison to B, you can see that the sodium channel is closed. It's not allowing in any sodium, which, if you remember, that's a positively charged ion, so we're going to depolarize our neuron this way. In B, acetylcholine attaches to that receptor, and we see that inner portion open up and allow in the sodium. So again, an ionotropic effect is just a flow of ions. And I'm going to talk about metabotropic effects for just a little bit longer because they involve this second messenger system. They're a little bit more involved. So we initiate a sequence of metabolic reactions. These emerge 30 milliseconds or more after the release of the neurotransmitters, and they typically last up to a few seconds, sometimes longer. So these neurotransmitters that influence mood, hunger, and things like that are more slow and long-lasting are having these metabotropic effects. And some examples, so serotonin, dopamine, norepinephrine, sometimes glutamate and GABA, but so they act differently depending on the kind of glutamate and the kind of GABA. And I've tried to remain really simple, and I usually do this somewhere with the neurotransmitters, but there are just different kinds of, so we'll talk about two different kinds of glutamate when we talk about learning and memory. There are multiple different kinds of dopamine, and I think they have the names like dopamine 1, dopamine 2, dopamine 3. There are different kinds of histamine, there are different kinds of serotonin as well. As I say, I like to work from pictures because I think it helps us to visualize how some of this is happening. So working from this picture, a neurotransmitter is going to attach to our metabotropic receptor. And what happens then is we see this bending of the receptor protein that's going through the membrane. And when that receptor protein bends like that, the other side of the receptor, we have this G protein that was attached, and that bending causes the G protein to detach, where the G protein is just this protein coupled to guanosine triphosphate, which is an energy storing molecule. And that G protein is going to act as a second messenger. It's going to take its energy somewhere else into the cell. One of the, I think it's actually your author who describes this kind of weird situation, but I think it's helpful. I'm not really sure that it's helpful, but if it is helpful, we can think of it kind of like I want to see the rabbits start to move, and I have my dog, and my dog is in a cage. This is a really weird analogy, but then my dog is in a cage, and I go in with a stick or something, and I rile up my dog and open the cage, and so that dog is acting as the G protein because now the dog is going to go upset the rabbits. Well, this is the energy of and what's going on within the cell is what we're going to call the rabbits in this case. I'm not really sure that analogy helps, but if it does, good. So the second messenger is then communicating with other areas of the cell, and it might just be this simple opening and closing of ion channels, just less directly. But sometimes we see an altering of the production of activating proteins, or activating that it activates chromosomes. So we see differences in gene activity and gene expression from some of these metabotropic effects. And this is more important for more enduring effects. So sometimes these effects last 20 minutes or so. Taste, smell, hunger, thirst, pain, where the fast timing, that kind of fast information processing, is not as important. And then we also see that this is important for many aspects of things like arousal, attention, pleasure, and emotion. So as you can imagine, things like serotonin, dopamine that are influencing our arousal level, our mood, our attention, our hunger, eating, and so forth, and sleep cycles. Those are metabotropic effects. So I'm going to distinguish now neuropeptides from other neurotransmitters. And your author does this really nicely in a table that I think puts everything together so clearly. Neuropeptides are often referred to as neuromodulators. There are other neurotransmitters that we sometimes call neuromodulators. So serotonin is a neuromodulator, but the neuropeptides are neuromodulators, and they're behaving differently than the other neurotransmitters. So when I talked about how the action potential affects the release of neurotransmitters at that presynaptic terminal, I was really talking about neurotransmitters in that case. Neuropeptides they behave a bit differently, so they're not synthesized in that presynaptic terminal for release there. Instead, they are synthesized in the cell body, and they start to travel down the axon. So we have them in the cell body, and when they get released, then they're mostly released from the dendrites, but also from the cell body and from the sides of the axon. And they're going to diffuse to a wide area of the brain, partly themselves diffusing out from the cell and traveling, but also they have this effect on their neighboring cells such that they are releasing neuropeptides too. So you can see why we call this neuromodulation. They are not released by the single action potential, as I was talking about the neurotransmitters before. This is when a neuron has repeated depolarization. So we see more than one action potential, and that's repeating. Then we release neuropeptides. Again, the duration of these effects can be many minutes instead of being more generally that fewer than less than a second or a few seconds or so. So one of the most common, or one of the ones that I talk about in more classes than other neuropeptides, are these endorphins, which endorphins really comes from endogenous morphine. They resemble those opiate drugs in structure. We actually discovered them with this logic of opiate drugs work. We must have some kind of chemical in the brain that is performing the same job and fitting into those same receptor sites, or we would not have those receptor sites. And this is where they came up with this as calling them endogenous morphine. They have an inhibitory effect, and they play a role in pain relief and response to stress. They also contribute to the regulation of eating behavior. So if you've when I was young, I was on the track team. And I really enjoyed my runner's high, I have to say. Those natural endorphins, that runner's high, is a great feeling as you start to have your basic pain, and you have this runner's high, and it feels you're relaxed and you just don't care so much about the pain, and you're also not hungry at that point. I want to go through this summary of drugs and their effects as briefly as I can while being as informative as I can. Starting from the top and then going down and moving kind of clockwise around. I'm going to talk about, again, agonists as drugs that are enhancing the effects of a neurotransmitter, and antagonists as opposing the effects of a neurotransmitter. So we're getting, if you look at the top, tyrosine, we're getting tyrosine, phenylalanine, we're getting that from our diet. And those amino acids are utilized for creating DOPA, which will then be used for creating dopamine. If usually AMPT, that drug that's mentioned up there, is used sometimes in research. That drug blocks that chemical reaction, so it does not allow DOPA to be created, and thus it is an antagonist for dopamine. If you've heard of people taking L-DOPA for their Parkinson's, that is the precursor to dopamine. It crosses the blood-brain barrier, and it can more directly be synthesized into dopamine. As we move on down, you can see it mentions MAO, the monoamine oxidase, which I just mentioned on a few slides ago. That enzyme is breaking down those monoamines, including dopamine, and so that we don't have an excess in the terminal button. So one of the kind of last resort antidepressants is called a monoamine oxidase inhibitor. It's inhibiting that enzyme, and so actually it is a dopamine agonist. Because that enzyme's not working as much, we can allow the dopamine to stay in those terminal buttons so that it can be used. If we continue kind of down where you see that reuptake, cocaine, methylphenidate, some of the antidepressants are blocking the reuptake. My memory is also that some amphetamines are also just triggering the release of dopamine. So all of those are dopamine agonists. If we look at the antipsychotics that are used by schizophrenics, or they prescribe to schizophrenics, what they usually do is they go into the receptor site of the postsynaptic neuron, fit into that receptor site. They do not alter the activity of the postsynaptic neuron, but they are blocking the dopamine, so they are dopamine antagonists. And then we also have cannabinoids, which can, so usually what I'm doing is I as the presynaptic neuron, I'm releasing the dopamine. I am allowing the dopamine back into my autoreceptors that basically tell me to inhibit further release, and cannabinoids are doing that same kind of fitting into those autoreceptors to inhibit further release of a neurotransmitter. So hormones are chemicals that are secreted by cells in one part of the body, and they're actually conveyed by the blood so that they can influence other cells in other parts of the body. And your author, I think it's your author that has this nice analogy of a neurotransmitter being like a telephone signal that we are calling, or we are sending the message to the specific neuron or neurons. We can have a little conference call, the neurons that we need to communicate to. Hormones are more like a radio signal. So we send out the signal through the body, and we can have an influence on whichever cells are recognizing these hormones, regardless of where they are in the body. And those neuropeptides, those neuromodulators, are more intermediate. I actually like the analogy to be more like the neuropeptides are like an FM radio. So if you're tuned to my channel and you're pretty close by, you can get my signal. And hormones more like an AM channel, or more like maybe even ham radio. So anywhere in the world that I want to send my signal, is if I'm on a certain radio station and you're tuned to my radio station, you can get my signal regardless of where you are in the world. Just talking in the body instead of the world. And your author gives this nice picture of the major endocrine glands. I feel like there's a lot to learn in this class, and I'm not teaching endocrinology, and the endocrine system is so complicated that for the most part, especially whichever one we get into of ingestive behaviors or reproductive behaviors, we'll talk about those relevant endocrine glands when they become relevant. But it's right now what you want to know about is what's going on in the brain. So I'll be talking about the hypothalamus and the pituitary gland as really the major control of this endocrine system. There are various types of hormones, and they are all these chains of amino acids. So protein hormones are longer chains of amino acids, and peptide hormones are shorter chains. Hormones attach to membrane receptors where they activate a second messenger. So they have this, they act like a metabotropic synapse. And the circulating hormones are going to modify brain activity, and then hormones secreted by the brain are going to control the secretion of other hormones. And I'm going to show all this. This is just a summary of what I'm going to show with some pictures here. So the pituitary gland is a small gland there at the base of the brain. It is attached to the hypothalamus. And there are actually two distinct glands of the pituitary, the anterior gland, or the anterior pituitary, which is this glandular tissue, and the posterior pituitary, which is neural tissue really extending down from the hypothalamus. So it can be considered an extension of the hypothalamus. So I start here with the posterior pituitary. And so these neurons in the hypothalamus are doing it's the hypothalamus that is synthesizing the hormones oxytocin and vasopressin, this antidiuretic hormone. And these migrate down to the posterior pituitary, and then they get released from there into the bloodstream. If we want to see what's happening in the anterior pituitary gland, the hypothalamus is usually is stimulating the anterior pituitary by secreting some releasing factor. So in my example there, that is thyroid stimulating hormone releasing factor, or releasing hormone, which then communicates to the anterior pituitary so that it secretes its thyroid stimulating hormone, which communicates to the thyroid gland in the body. The thyroid gland releases its hormones, and as its hormones, so all of those are excitatory, as its hormones are released, we get this inhibitory message, or this negative kind of feedback loop back to the hypothalamus, telling the hypothalamus to stop its secretion of that releasing factor. The hypothalamus does have a couple of inhibitory releasing factors communicating to the anterior pituitary. But again, for right now, it's just how the basics of the system, that it goes hypothalamus to the pituitary to whatever gland in the body that is going to then release the hormones in the body that then have this negative feedback back to the hypothalamus. So we've talked now about ionotropic reactions or receptors, where we need that faster transmission of information, as in vision and hearing, and metabotropic reactions or metabotropic receptors, where instead of just letting the ions through and changing the voltage of the cell, we're going to do something. We're going to communicate to a second messenger. We might have some effect on genetic expression or the metabolism of the cell. We might also let in ions, but those are slower reactions and longer lasting reactions. Well, we also have a few special purpose synapses that convey messages electrically with this really fast transmission, faster than chemical transmission. And this is important when we have two cells that need to behave in exact synchrony with one another. Your author gives the example of some of those cells that control rhythmic breathing. So we need to breathe, those cells need to be doing the same thing at exactly the same time. And sometimes I see that prey animals, their sympathetic nervous system, that fight or flight response, some of those cells have this kind of electrical synapse that allows them to move very quickly. And again, the cells behaving with exact synchrony. And this is these electrical synapses. So this is where the membrane of the presynaptic neuron is going to come into direct contact with the membrane of the postsynaptic neuron. And you can see it in the picture, and at the bottom there you can see these pores, those channels line up. So basically the presynaptic neuron is sharing its ions with the postsynaptic neuron. So this is just a quick electrical change. And this is happening at what's called a gap junction. Okay, where those pores are lining up and those neurons are communicating is the gap junction between those two neurons in our electrical synapse.